You have just been diagnosed with open-angle glaucoma. I know that you may be experiencing feelings of overwhelm and concern, but today I'm going to be explaining to you more about what glaucoma is and present some cutting-edge research that you can potentially contribute to help cure this disease and reverse the irreversible blindness. Let's begin with an understanding of how vision works. Normally, light enters your eye and it travels through the lens, the clear part, until it reaches the very back of the eye, the retina. In the retina, there are a lot of photoreceptors, so they accept the light and they turn it into electrical signals that can travel through the optic nerve to the brain, where the image is finally processed. As you can imagine, this optic nerve pathway is crucial for the image to actually be translated into something that can be understood. And so, in the presentation today, we'll be focusing on the optic nerve. And in particular, one of the cells that makes it up, the retinal ganglion cell. As we can see here, the ganglion cell is in the very back of the eye, and it's made up of a cell body, the circular part, and axons. The axons actually make up the optic nerve. This is so important because it's the direct pathway from the eye to the brain. An important thing to note about RGCs is that they cannot regenerate if they're damaged or killed. It's super important for our vision to maintain them. Unfortunately, in glaucoma, this is not the case. Glaucoma results from increased intraocular pressure. So increased pressure inside the eye actually damages the optic nerve and lowers this connection. Additionally, these RGCs die off. In diagram A, we see an eye that has normal pressure and many RGCs, these red dots. In diagram B, after 2 weeks of increased pressure, there's already less RGCs. And in diagram C, after 4 weeks of increased intraocular pressure, less than half remain. As you can imagine, this is detrimental to vision. Now, you may be kind of confused, wondering, how do I have glaucoma? I didn't notice anything. Well, this disease is actually deemed the silent thief of sight. You have to lose at least 30% of the RGCs before there's any detectable change in vision. Additionally, the RGCs that die off first are those that affect your peripheral vision from the side, so you may not notice anything. But as the disease advances, it leads to tunnel vision, like you're looking through a telescope. And as we said, because the RGCs cannot be regenerated, once this vision is lost, it is irreversible. There are some treatments if glaucoma is caught early to stop further vision loss, such as eye drops and surgery that reduce the pressure. But again, the vision that was already lost is gone. Right now, this disease does not have a cure. But today, I'm going to be sharing with you some exciting research. Making the front cover of Nature article published just this past December in 2020, Dr. Claire and his fellow researchers at Harvard University published an article entitled "Reprogramming to Recover Youthful Epigenetic Information and Restore Vision." Now, this sounds like a lot, but I hope the idea of restoring vision sounds exciting to you, and I'll be breaking down some of the experiments and the promising results. To understand this work, we first need to understand what Yamanaka factors are. These are OCT4, SOX2, and KLF4. Now, don't worry too much about the names. We're going to call them OSK because they're always together. And basically, if we isolate cells that are already cultured, they already have their function, and add OSK, which are essentially reprogramming factors, the cells become pluripotent stem cells. This means that they can differentiate into any type of cell. We're essentially stripping all of their old knowledge and letting them form new connections. So in this diagram here, it could become a blood cell, a gut cell, a muscle cell. And so Dr. Sinclair and his fellow researchers decided to try activating OSK in the retinal ganglion cells so they could essentially reset the connections, reversing the effects of injury and hopefully restoring vision. In the first experiment, what they did is they injected the OSK into the eye of a mouse through a virus, AAV2. Then they performed an optic nerve crush injury. As stated before, in glaucoma, the optic nerve is damaged, so we needed to simulate that here. They let the OSK be induced for 12 weeks after the crush, and we wanted to measure the number of axons. As remember, that's part of the RGC that makes up the optic nerve. And so by inserting CTB, the axons glowed. And here are our results. On the top is a mouse that had no OSK, and the bottom is the one that had OSK induced for 12 weeks. The crush was performed right here. And as we can see in the top, we see the bright lines representing the axons before the crush, but in the mice that had no OSK, there are no axons after. They all died off. But in the mouse eye that had the OSK expressed for 12 weeks, it is so much brighter. These axons had a regenerating and survival rate. This means that the mice could potentially form new connections between the eye and the brain, and vision could be restored. Now, the experiments don't stop there. They did an experiment to simulate glaucoma even more closely. This time, to damage the optic nerve, they increased the intraocular pressure by injecting microbeads into the mouse's eye. Of course, the microbeads decreased the axonal density and the number of RGCs, just like in glaucoma. And in the experimental mice, they injected OSK through this virus once again. Of course, the OSK increased the axons like we just saw. They also tested the direct effects on vision. To do this, they used an optomotor response. The mouse was placed on a pedestal in the center of 4 computer monitors. These monitors had a striped black and white pattern, and they rotated in a circle around the mouse. They began with very thick lines, or a low cycle per degree. And the researcher tracked the reflexive head movements. So positive tracking is when the mouse moved its head in the direction of the bars. And if there was positive tracking, then they increased the cycle per degree, or made thinner lines, which are harder to track. And they kept on doing this until the threshold was reached, until there was no more positive tracking. And here are the results. We're going to be focusing on the treated group of this graph. The gray bar here represents mice that were injected with just a saline solution. So there were no microbeads, no increased pressure, and so no damage to the vision. And of course, they had the best vision. Just to note, the y-axis here is optomotor acuity, so the vision sharpness, or cycle per degree. Better cycle per degree, better vision. The orange and red bars we're going to look at together, as microbeads were injected, but no OSK was induced. Here there was saline, and here the virus was injected, but the OSK was not expressed. And of course, we can see that this had the lowest vision because the RGCs were damaged and there was nothing to regenerate the axons. But the exciting, promising results are this blue bar here, the experimental mice that were injected with the microbeads and had the OSK expressed. As you can see, the vision actually increased. It's higher than those that just had the microbeads and is climbing towards the bar that had no injury at all. 50% of the visual acuity that was lost from the increased intraocular pressure was restored. And this is so promising, meaning that there could be a possibility to cure glaucoma. So, to recap these experiments, OSK expression had a regenerating effect on these retinal ganglion cells in mice. The axon density increased, as we saw in the first image, which with all those glowing lines, and vision was restored. They could see higher cycle per degrees in that optomotor response measurement. Now, the work does not stop there. We see that OSK has a regenerating effect, but Dr. Sinclair and his fellow researchers want to understand how these cells actually encode the information that allows OSK to work. Once they understand how these cells have this information, they can spread their research to other cells throughout other parts of the body and induce OSK to cure other aging-related diseases by essentially resetting the cells and allowing it to form new connections. But our first stop is focusing on curing glaucoma. And excitingly, in 2 years, there are human trials planned. And if you contribute, you will help with all this funding to make these trials possible. And with that, I hope that you'll consider donating and supporting the Sinclair Lab at Harvard University. Thank you for your time and attention. And if you have any questions, I'd be happy to answer.